On the coast, not in Hattiesburg. I'm at the Gulf Coast Research Lab in Ocean Springs, and I'm stalling for a presentation because I asked to change it last minute, like I usually do. My work involves a lot of migratory fishes. We do a lot in estuarine and in the Gulf of Mexico. But I've been working with Gulf sturgeon in the Pascagoula with some of the folks in this room since 2015, and we started working on the Pearl River in 2020. Robbie had a nice little introduction to sturgeon in his talk, along with some of the impediments that are along the river itself. So I'm going to jump into my talk a little bit. So Gulf sturgeon are an anadromous species, which means that they spawn in freshwater habitats and they derive their energy from estuarine and marine systems. Gulf sturgeon occupy well, look, there's a nice little Gulf sturgeon and all my social things. Now I'm going to get into the first bit. You'll see from the talk sort of the range of work that we have for telemetry programs, monitoring different fish species, and it could not be done without a great group of people. So my current lab is pictured here, along with former members, and these two gentlemen, one of which is in the room, my postdoc advisor, Mark Peterson, and a much younger looking Todd Slack, have been integral to helping us understand what's happening. Wow, it's a lot louder over here. With Gulf sturgeon in these systems. So their pattern of anadromy, okay, I got it wrong. Got a lot of words. So Gulf sturgeon are sometimes called a dinosaur fish or an ugly fish. The modern form of sturgeons sort of diverged from the paddlefish lineage during the Jurassic period, so you should have seen these guys in Jurassic Park rather than some of the dinosaurs that you actually saw. The Gulf sturgeon is a species of the sea sturgeon clade, so that includes the European sturgeon, Accipenser stereo, the Atlantic sturgeon, Accipenser oxyrhynchus, and the Gulf sturgeon, which for a while was considered to be a subspecies of the Atlantic sturgeon. Now it's Accipenser desodoi. Their range is found in large coastal rivers within the Gulf of Mexico, presumably no longer interbreeding with the extension of the Florida Peninsula. Gulf sturgeon are found currently in 7 drainages from the Pearl River where we are through the Pascagoula all the way to the Suwannee River in Florida, and they've been extirpated from the Mobile River system, which is why you don't see them on the map. They can be genetically separated roughly into 2 population units, an eastern population unit, which is rivers that are east of Mobile Bay, and a western population unit, which are the Pearl and the Pascagoula. And since a running theme of this has been, this is the old talk, parasites, there's actually a species of external parasite, Accipenser stitzostetii, that's found Argula stitzostetii. It's a branchiurin and external parasite, only found in the eastern ones. And what we have in the west is Argulus flavescens. So this sort of unknown about what the population genetics and how the mobile systems are related to the rest can probably be resolved by looking at what species of Argulus is found in that system there. Robbie went through the impediments that they were faced on the Pearl River, but across their range, changes in habitat use through deforestation, river impoundments, as well as a brief but really intense commercial fishery for sturgeons around the turn of the century is what led to the major decline in their populations. So for the Pearl River, there's no known harvest numbers until about the 1940s. And even then, it's really only about 500 pounds or so per year out of this system. But our nearest neighbor, the Pascagoula, in 1902, you can see that there was a staggering 24,000 pounds of harvest that came off of that system once we started recording the collections. Since that time and through the 20s, 30s, 40s, and 50s, it sort of dropped down quite a bit, down into that 1,000 range. And then when the harvest ban went into effect in 1974, we've still seen sort of these stagnated populations. So it's like the 1-2 punch of being extracted, especially when you're fishing for the females that are producing the young for caviar, and then changing their habitats downfield. In 1991, they were listed under the Endangered Species Act, and they're one of the few species where critical habitat for the species wasn't listed until 2003. So really their populations were historically declined, and we've yet to see them truly recover. This is largely related to that anadromous life cycle. So they can be found feeding in the Gulf of Mexico and within Mississippi Sound, Lake Pontchartrain, usually between October through April or May, depending on whether or not they're a spawning individual. I like to say that they leave our rivers around Halloween so that they can go trick-or-treating and go eat. What they like to feed upon, I know it's close to lunchtime, so I'm trying to stave you off from leaving real quick. But what they like to feed upon around the barrier islands are really yabbies, ghost shrimps, callianacids, all the same word. So they slurp up these little high-density, energy-rich prey items using a tube-like mouth right out of the infauna. These guys, they filter feed. So menhaden are also a filter feeder that has a lot of oil and has a tremendous amount of benefit to our estuarine systems. They're functionally feeding on the mullets of the stand bottom by feeding on these things. And they can feed on hundreds of these in a go along our barrier islands. When they're smaller, younger, juvenile fish, they're feeding on chironomids, usually within low salinity and somewhat freshwater locations. Where they spawn, I used to make jokes about the snow-capped mountains of Mississippi, but then we got 7 inches of snow a couple weeks ago at the house on the coast. But they spawn upriver, usually over gravel-type substrates, hard-bottom substrates. They have adhesive eggs, so sticky eggs. They like flowing water, rich of oxygen, so that they can develop, and free of sands and silts, so that they don't suffocate those eggs. Sturgeon then Gulf sturgeon do this weird thing where instead of returning back out to marine systems to then go back and feed like most other sea sturgeons do, and in fact a lot of anadromous fish do that are semiparous or that spawn multiple years in a row, they undergo a period of holding in the lower portions of rivers. So you can find them in like low stream velocity locations, areas that have some good depth, or perhaps areas that have a good number of logs and snags where they can stay on the downstream side of it to conserve energy, because during this time they're apparently non-feeding. Some of the fish that I've looked at in Florida that were killed fish actually have food items occasionally within their intestinal lining, but any of the necropsies that I've done for Mississippi ones have all sort of been empty. So they're really weird, this sort of weird pattern, right? Then October comes and they move back out. For the young of the year, when they're produced in these locations, they have their periods, they have distinctive sort of black and brownish eggs. These are a couple that we found off the Bowie River for the Pascagoula, around the only known spawning site in that system. They then hatch out, they go through a period of time where they're sort of planktonic before settling down, changing into the black stage where they're basically drifting downriver as looking like a little bit of detritus or leaf material. They then enter into a bicolor stage, which is right about the time they're starting to kiss saltwater-ish for the first time, and then running way quickly through 7 slides at one time. Go back. Okay. Until they're about an age 0 or an age 1, truly entering that stage, and then they'll return back into freshwater each year. So they undergo this pattern of feeding in estuarine conditions, during the winter time, and then moving back into freshwater habitats, regardless if they're spawning or not, to stay in the rivers before doing it again. So undergo what we kind of dub as a trophic ratchet. Go down, get fat like me, and then go back up and try to conserve as much of that as possible while you're fasting. It's a pretty extreme diet. Don't recommend it. All right. So what does my group do? So where sort of our goals and objectives, I mentioned it before, a little bit on understanding their movement patterns and migrations, but also using this information to help understand population demographics to aid in recovery goals for the species. So within the Pearl River itself, again, we've been doing work in this system since 2020. Before us, Todd Slack's crew, Kayla Kimball, and Ashley Bear, who's in the back, did some work out of Baton Rouge field office as well. But our catch numbers sort of look here. I don't know if y'all can see on this side with this slide, but generally speaking, we're catching fish that are not true adults in the system. So right at about 125 centimeters fork length, we're at about just over 4 feet. That's when we consider a fish to be an adult. Some of this is related to sampling bias. We are targeting for one of our projects juveniles, but for 2 other projects, we're also targeting adults, so using large mesh gear. And you can see that our capture rates are really low for adults in this system. We can use this, Will mentioned it in his talk, but mark recapture data, getting resites or recaptures of the same individuals are really valuable. You can use this over a period of time to try to build population estimates. And so that's what we did using Cormac Jolly Sieber model, which is an open population model for Gulf sturgeon that are greater than 110 centimeters in fork length, so on the edge of being adults, but it allows them to then grow into an adult stage over time. And so our first estimate in 2021, we had 86 individuals. This is the 95% confidence interval at the end. 2022, 122, 2023, 128. So pretty low-ish numbers for an adult group, especially when you're looking at juvenile catch numbers that are more than 200 over a 4-year period. So it seems like either I'm really bad at catching them, which is part of it, or we're having an issue in getting those small fish to being adults within the system. We see a similar trend in the Pascagoula as well, although we have way fewer juvenile fish and a lot more subadult or sort of intermediately sized fish. We also sort of care a little bit about age structure, specifically for younger fish. So if we take a look at the distribution, the age bands of data, that includes fish that Todd Slack had worked on for part of his postdoc with Steve Ross's lab out of USM, as well as data that was after Hurricane Katrina, but and it sort of also occurred around the oil spill. We have sort of that same shift, right? A lot of younger fish within the system, not a lot of those old fish. To age them, we do it non-lethally. So for a lot of teleos fish, your normal bony fish, you can pop their heads open and pull out their ear bones or otoliths, section them, and then count them like rings of a tree. We don't want to do that to a sturgeon, and also their otoliths are made of a different carbonate compound. But what we can do is we can actually take a section of their pectoral fin, because it's a hardened structure, and from there we can count annuli associated with those rings. So each of these dots designates an age of that individual, and we can do this non-lethally, so we can put the species right back in. From generating growth curves for the western population segment, which is what these are here, we can see that for a mature fish at about 125 centimeters fork length, they're right in that 7-8 zone. And again, we're seeing relatively few individuals that are larger than that, which could cause some concern because usually females don't develop until about 8 or 9. So, yeah. The other side that we're interested in, I mentioned that we had a juvenile project. So one of my PhD students is looking at comparing growth of Gulf sturgeon across their entire range. So from the pearl all the way over to the Swanee. And so what we have here are the fork lengths of age 1 individual, so fish that were that have been around for a year and some change. From the pearl here in this red color, the Pascagoula in whatever color that is, yellowy green, and the Pensacola Bay systems, we group 2 rivers together for ease. We can see that the westernmost rivers overall have lower growth or lower sizes at age 1, but by age 4, there's no difference in those lengths. So our fish might be starting off by not reaching size as fast as those to the east, but by the time they reach about age 4, we've caught back up again. So a little bit of good news there. The bread and butter of the work that we do in my lab is associated with acoustic telemetry. So let's fix that from Mississippi to some brown water. So the way this technology works is that you have these hydrophones that are listening for a particular code that is coming out of a transmitter that we put in the fish. The battery lives on these range anywhere from about 30 days for a really small tag to go into small fish, up to 10 years for larger tags that we put into right around 1,000 millimeter, 100 centimeter fish. So we can get a lot of data for a long period of time using those tags. What they have to do is you have to have a fish that swims around carrying its little surgically implanted tag. It gives a ping, so it gives us a date timestamp, as well as the individual sort of serial number for that fish. And then it moves along and it can be detected in a different location with a new date timestamp, so we can piece together its movements. But we can also use that to get virtual recapture, so we don't have to physically recapture a fish. We know it's alive if it's swimming around. Well, so long as it wasn't eaten by a shark. So, to do this, we have to deploy an array of these receivers. We use a lot of different mooring methods to try to attach them in different areas. But on this map, you can see where we run. So we're working from where we are now all the way out to the Barrier Islands, collaborating with Mississippi Department of Marine Resources to do some of our offshore work. And then we're doing the same thing throughout the Pascagoula. So we have a pretty extensive array. And it takes a lot of time. But what you can enable are movement tracks. So you're going to see a bunch of lines. They're all in the rivers now in the Pascagoula. And they come out and they make use of the Barrier Island complex. Those that are in purple are juveniles, less than 89 centimeters. Those that are in yellow are subadults, and those that are blue are individuals that are adults. So they're the ones that made a spawning run on that video. So if you notice, all these fish are in the mouth of in the lower portion of the Pascagoula River. As we move around, we're now in October. They're around the Barrier Islands. Most of the juveniles hanging out around the estuary, subadults pretending to be a big dog bouncing around, and the adults making spawning runs. Here's an individual that came all the way up to the reservoir in the Pearl. That fish was originally tagged in the Pascagoula, not in the Pearl. So, one of the questions that we then have, and maybe we shouldn't have been too surprised considering I'm standing next to the geologic map of Mississippi, but one of the questions we want to know is, what does the substrates look like available for Gulf sturgeon spawning habitat in the Pearl and in the Pascagoula, so we can help narrow down potential areas where they might be spawning. So each of these dots is one of those hydrophones receiver arrays. This is in the Bowie River, a tributary of the Leaf that goes into the Pascagoula. The reason why it's not outlined for where the river is because it's outside of federally designated critical habitat for Gulf sturgeon. And the same thing is true on the top side when you're running here's into the Chunky River. So we used just recreational grade side scan sonar, like a Hummingbird unit. Went down at bank level flows to try to, well, we did scans of of all of these stretches of the river in the Pearl and in the Pascagoula, as well as their tribs in the Boca Chica. We didn't do the strong because of issues with passage at the time and log jams. But what you can see here is colors that are much higher in yellow are fines, so those are sands. So this is the amount of sort of sands and muds that are throughout most of the pearl, and you can see that largely it's in that yellow-orange color, right? So a lot of sands throughout this system. So we do the opposite side then, and we want to find areas that have nice yellow on this particular map, which is where we have a lot of hard bottom, a lot of gravels and hard returns. And so what you can see is sort of through the theme of most of the talks, it's that Byron Bridge down to the mouth of the Strong, where in the Pearl we see most of that hard bottom habitat and potential spawning locations then for Gulf sturgeon in this particular system. If you look at the Bogachita, it was some of LDWF and Baton Rouge's work had found adults relatively high up in the Bogachita, and I think, correct me if I'm wrong, but above there's a big adult in Franklinton, Louisiana, that's posted on somebody's wall or something. But there's not a lot of potential spawning habitat in that system. And realistically, for the Pearl compared to the Pascagoula, there are a lot fewer locations where you have lots of good spawning habitat. This is pretty much the only stretch where it exists. In the Pascagoula, even though it's not really highlighted, this is the Bowie River where there's a historically aggregate mine site. This is the location where we've confirmed spawning, both with my lab as well as some of the work that Todd and Steve's group had done. And then we've noticed different locations in the Leaf, as well as going all the way up into the Chunky for the Chickasee, where spawning habitat is for Gulf sturgeon. And their telemetry tracks for the Pascagoula follow. For the Pearl, since this is Pearl River Day, these are some plots that demonstrate fish that are found at least around river kilometer 40 within the Pearl, and then moved above where the Bogachita sill is, or sill 2 associated with the navigation canal, or Pools Bluff sill, which was also put in place for the navigation canal, as Robbie mentioned, in the 50s. They're right about the same river kilometer, so for my ease and brain, I just made the color the same. So that's right about river kilometer 100. Anything that has a BC are fish that moved into the Bogachita rather than moving into the Pearl, but you can notice that they're not going very far. There's, again, not a lot of potential spawning habitat, and they're only going about 170 river kilometers in that system. So really just above that zone a little bit more. There's a tubing center around that area. There's some really poor quality habitat, but a lot of riprap. So we're presuming that they're maybe using that riprap location. When it comes to the Pearl River, you can see a couple of individuals. So this is that one that 3 years in a row made it all the way to Ross Barnett Reservoir. This is a male fish, but it spent most of its time right in that stretch between the Strong and Byron. Same thing is true for this particular female and this particular female. They all made it to about that same section, and then they stopped their run. So genetically, we know that they're female. Brian Preiser's lab at USM in Hattiesburg has been able to not just do the population genetics, but also to genotypically sex those. But what's sort of disappointing, right, is that the vast majority of the fish that we've tagged are not going above Pools Bluff. They can pass it at about the river gauge at Bogalusa, at 13 and a half to 14 feet. Gulf sturgeon are capable of passing over that sill, but we're seeing very few fish try to even make it. So these fish that are here at the sill at these times, they can pass if they wanted to. They just didn't. So it raises another question in terms of all these juveniles that we've seen now, started with Baton Rouge's work, continued with our work in the Pearl River. Is it possibly that they're just being produced right there? So the adults are not going, not very many anyway, adults are going above that system, but a good number of them are staying. If it wasn't for the plasticity of sturgeon in general, there would be no lake sturgeon throughout the Mississippi River system if they didn't make use of spawning at the footprints of dams and sills on those rivers. Here's a side scan sonar image. There's the sill berm here. This is a concrete apron. And if you're ever sort of within that stretch, or you've walked down to that location, you'll notice that there's a lot of, obviously, water is falling, so it's touching air, coming into the system, being oxygenated. Because blowhead dams are also known as drowning machines, they have this period of water overflowing. So presumably helping keep, we sort of see it here, keeping those fine sands and silts out of that location. And so sort of what I'm guessing is that a number of those fish that are not going above but present during the spawning season are making use of that system and being produced. Dr. Peter Allen at Mississippi State is using our fin spine samples along with Brian Kreiser's genetic data so that we can start to try to fit this story together using the microchemistry of the water. So depending on where water comes from, it carries the signature of the land that's with it. And what we think that we've seen so far is that the vast majority of juvenile Gulf sturgeon, age 1 and age 2 Gulf sturgeon that we've seen since 2020, all have, or the majority of them have a signature that's not too terribly different than the signature of being near saltwater. So this is river kilometer 100. The water chemistry is pretty similar until you actually make it into saltwater. So it's starting to help line up the evidence that this is probably where they're spawning. Now let's shift into the marine realm. So talking about where Gulf sturgeon go throughout the Gulf of Mexico as well as Mississippi Sound. My lab, again through collaboration with Todd's group, has been very fortunate to try to address questions related to habitat restoration. So there is some concern that creation of oyster reef habitat, or at least putting the limestone material down within the soft bottom habitats of Mississippi Sound, since all of this is designated, these blue outlines here, all of this is designated as critical habitat for Gulf sturgeon, is at a potential detriment as they're losing open bottom feeding areas to the creation of reefs. So we have a collaborative project that's sort of in its last year where we've established very fine scale arrays where we can get individual positions of Gulf sturgeon around open bottom, so areas we're not touching, around areas that were open bottom but are now oyster lease habitats, so areas where we put treatments out with crushed limestone in different patches, and then areas where there's naturally occurring reefs in both the St. Louis Bay system, which is shown here, and the Pascagoula River system. For the purposes of time, I'm just going to show you what we've seen for the Pascagoula side, because it's well, one, it's a lot cleaner, and two, we're a little bit further along on the analyses. So if we look at the total number of detections that we have around for Gulf sturgeon, they're primarily associated with those oyster reef locations and not the locations that are open bottom. What we also see is that the locations where oysters are, we have more percent sand than we do percent silts and clays. So sturgeon are presumably identifying locations that have higher sand. One of the reasons why you might postulate that sturgeon would avoid an oyster reef is they have a suction tube mouth. It's not very efficient for slurping things around the very jagged, ragged edges of an oyster reef. But what's really cool that you can see on this confetti plot is that sturgeon can make use of resources from an oyster reef that aren't directly on the reef themselves. So what we hypothesized in the beginning is that Gulf sturgeon are probably making use of what we would call a halo effect. As oysters are feeding and pooping, they're getting rid of feces and pseudofeces, and they're going right into the sediments that surround them. They're also changing the way that water flows in those areas, presumably also identifying locations where they're not just going, the young oysters aren't immediately going to sink into the mud and get buried, because then they can't persist. So hard bottom locations where there's more sand and more reef material is right around the time when you see all these Gulf sturgeon hanging around the periphery. So making use of those locations that are on the outside or the halo of reefs, rather than actually going across the reef complex themselves. Another restoration project that we've been involved with is that of Ship Island, Mississippi. It's the Corps' now second largest, but at the time largest restoration project that they undertook. So Ship Island was separated off and on prior to Hurricane Camille. But after Hurricane Camille, the island never came back together again between an east and a west ship. And then after Hurricane Katrina, it was widened to the point of basically no return. The sediment budget associated with longshore drift was never going to be able to, within a timeline meaningful to us and meaningful to the shrimp industry, be able to fill in that area. There was some concern about the potential for saltwater inundation then into the sound. So they undertook a project before I started working with Gulf Sturgeon, doing pre-restoration monitoring, and then we've been able to help carry out the post-restoration monitoring once they filled and united Ship Island back to a singular island. What we found is that Gulf Sturgeon from both the western populations, so from the Pearl and the Pascagoula, as well as Gulf Sturgeon from the east, made use of the barrier island habitats. This is something that was at least first documented by Steve Ross's lab early on, trying to refine critical habitat for Gulf Sturgeon. But Mark Peterson, Todd Slack, and then master student Paige Vick really showed that at that time in 2012, 2014, the first couple of years of pre-restoration monitoring, there were a lot more eastern fish than western fish using that section. Part of that's related to the current tag pool that was there for western fish, but you can also see that overall the fish that were using that island from the east tend to be larger in size. One of the advantages of migration is that larger body animals should be able to migrate further, and so we sort of see that with our larger eastern fish even persisting in a post-restoration time. This purple color is the pearl, so now that we're in post-restoration monitoring, you'll notice that juveniles, which is what this bottom dash line is, are not making use of the area, but subadults and adults are making use of the barrier island complex from the Pearl and the Pascagoula in relation at least to the tag pool, pretty equal numbers. So it's good to see that the resources that restoration of uniting the island hasn't really modified or changed how Gulf Sturgeon are interacting. And actually, we think for a shark species, it actually benefited quite a bit. If we look at then the duration, so when do we see Gulf Sturgeon first arrive? That's what these sort of box plots are here for when they first leave the islands or last are found around the islands. So early October through really the end of May, across all of them, pre and post restoration, the tag numbers change, but the fish are showing up at about the same time and they're staying for about the same time. So again, more documentation that activity hasn't changed the way that sturgeon use the habitat. And then finally, before I stop talking at you, we've also been involved in trying to understand what's happening with Mobile Bay. As I mentioned, it's excluded from designation of critical habitat as it was perceived that Gulf sturgeon were extirpated from that system. And so one of my former master's students, Elizabeth Greenheck, made use of a receiver array that a collaborator at Dolphin Island Sea Lab, Dr. Sean Powers' lab, had established looking for movements of southern flounder and red drum. But what they ended up detecting a lot of was Gulf sturgeon. And so we see Gulf sturgeon even from the Pearl, moving all the way into Mobile Bay, going into the mouth. And we've had genetically assigned Pearl River fish making it almost as far as Claiborne Lock and Dam within Alabama. So it suggests then that within that system, we have a lot more sort of overlap in where our fish are running to and from. We've also seen fish from the Choctawhatchee make it there, but a large number from the Pascagoula, sort of the nearest neighbor to that system, making even the same types of runs into that river. So it's really cool stuff, and it's been brought up recently, at least in the lawsuit against, or potential lawsuit against the Corps, for best dredging practices in the bay, because there's a lot more sturgeon using that system now than we previously thought. This is a salinity map of bottom salinities within Mobile Bay. This is heading into Grand Bay, the area of Grand Bay. And so one of the reasons why we think Western fish are using this system more than fish from the east is that we have a lens of fresh water that extends into with the exception of the ship channel, that extends through this portion and pushes in to Mississippi Sound. So as our fish from the Pearl and the Pascagoula are running out of their rivers, they're touching that Mississippi Sound estuarine water, and it's energetically favorable for those fish, or at least more, to not have to change their osmoregulation capacities as they then move through this low salinity water into low salinity habitats. For the eastern fish, from the Choctawhatchee and from the yellow and Escambia, we usually see those fish come in a bit later, and we see them using the channel as it comes in. So these fish have to leave the Florida Panhandle, go out of estuarine water into fully marine water, and then move back into estuarine locations. So making use of a deep ship channel where you have persistent high salinity bottom water is likely a lot more energetically favorable for them. So it's been a pretty cool test case, and she's going to expand on that for some of her PhD work. All right, with that, my lab has been super lucky. I thank everybody who is up here, agencies, speakers that you've seen or will see again today. And with that, I'll take any questions. Also, go Birds. Yeah.
The yellow pesticides they're using 12 pesticides or the riprap on the side?
So to be honest, I don't know. I think that during that period, they're probably making use of both. When we've done the scans, when we've been able to pass it, we've actually seen some large-bodied fish in those locations. It's hard to know if they are sturgeon or if they are paddlefish or if they're an alligator. I presume they're not an alligator gar. But we've seen it sort of through both of those areas. In other, like on the Missouri for lake sturgeon, it's usually more towards where that concrete apron comes down going to the trough, rather than it is towards the sides where you've got that apron and riprap material that's better established. And there's usually gravels that are associated in those spots, and they don't really get buried by anything.
After the questions, I wanted to get with you because we collected data with Louisiana Department from the late 90s going into 2007. I had that data.
Yeah, sounds good. Yeah.
You got a shortage in hard bottom in the Pearl. Strong has a lot of hard bottom. Historically, we had big fish that went way up to down 49. And what changed in there is that totally secret crab.
Yeah, so when we've tried to scan the strong, there's been the mouth of that system, and Calvin, Robbie, and most of y'all that get quicker access to it than we do driving from the coast could probably tell me I'm an idiot. But there's a relatively large sandbar complex that kind of changes through that area. And depending on the flows of the time, when we had really good flows and we had a receiver further up around hard bottom in the strong, we had no detections of any Gulf sturgeon in that system. Then once we started having passage issues ourselves, because we don't have a jet boat to be able to get through that system, we've even stopped seeing detections associated around the mouth of the strong. One of those years, it's because the receiver, the whole thing got sedimented in, it got buried. But once we modified it, we're not seeing them really go into the mouth of the. None of the tagged ones. That's always the caveat. None of the tagged ones are going into that system.
Historically, they did.
Yeah.
Big fish.
Yeah, I mean, and that has been shared with us, and we were hopeful that we would be able to see it, but in the 4 years that we've been doing it, we haven't. So, all right.
If you you so much, Michael.